Namaste viewers, this is Dr. Pratibha KS from Keli Society's S. Nijringappa College, Rajaj Nagar, Bengaluru. Today I'm before you to discuss Biotechnology Theory Paper 6, Immunology and Animal Biotechnology. Okay, I request you all to sit with one pen and notebook so that you can make a note. If I'm going very quick, you can pause this video and you can make a note. Okay. So now let us go through the fifth semester B.Sc examination, December 2018. Paper code BTP 503, Immunology and Animal Biotechnology. Time 3 hours, maximum marks 70. Okay. Section A, write short notes on the following. Okay. Here 5 questions will be asked. Each 5 question carries 2 marks, so total 10 marks. I request everyone to kindly subscribe if you have not yet subscribed this channel, and after watching this video kindly like this video if you like and share this video to others. Let us go through the question number 1, tissue extract. Tissue extracts, for example, embryo extracts you can take here, okay? Extracts from tissues such as embryo or liver, spleen, leukocytes, tumor, bone marrow, etc. are also used for this culture of animal cells. Okay, that is extracts from some of the tissues such as embryo, liver, spleen, leukocytes, tumor, bone marrow can also be used for the culture of animal cells. You can write it over there. Where embryo extract of some of most common use is embryo extract. Tissue extract should be used before a week or stored at 27 degree of Celsius. Okay. Now let us go to the question number 2. ELISA stands for Enzyme Linked Immunosorbent Assay, is a method of target antigen or antibody capture in samples using a specific antibody or antigen and of target molecule detection or quantification using an enzyme reaction with its substrate. In ELISA, various antigen antibody combinations are used, always including an enzyme labeled antigen or antibody, and enzyme activity is measured colorimetrically. Okay, the enzyme activity is measured using a substrate that changes color when modified by the enzyme. Light absorption of the product formed after the substrate addition is measured and converted to numeric values. Depending on the antigen-antibody combination, the assay is called a direct ELISA, indirect ELISA, sandwich ELISA, and further competitive ELISA. Okay, for 2 marks, this much information is more than enough. Okay, if I'm going very quick, you can pause this video and make a note of these answer. And also you can go through the uh, slide number. The next question is HeLa cell lines. Okay, HeLa stands. Henrietta Lacks a lump in her abdomen was observed where she was diagnosed and treated for cervical cancer. That is adenocarcinoma of the cervix, a particularly aggressive type of cancer was observed in her body. Okay. They are immortal, that is to say that they were defined normal mechanisms of senescence by acquiring certain mutations. Following are some obvious features that HeLa cell lines differ from cancer cells. HeLa cells have anywhere from 76 to 80 total chromosomes, which is different from other normal cells of total 46 chromosomes. As we all know, HeLa cells have the ability to contaminate other cell lines. Due to its contaminating feature, many projects or studies should be marked as inconclusive or invalid. The telomerase, which allows for addition of sequences at the end of chromosome, is active during the process of HeLa cell division. So, DNA will not be damaged and cell does not die. Although there are some cancer cells which active tumor telomerase yet is likely to be effective in HeLa cells particularly. Okay, this is about for HeLa cell lines. we can write this particular answer for 2 marks. Hills and lines, very commonly which is used in the animal tissue culture. The next fourth question, B lymphocytes. Okay, B cells or B lymphocytes are lymphocytes that originate and mature in the bone marrow. The antigen receptors of B cells bind to freely circulating antigens. When B cells encounter antigens that bind to their antigen binding sites, the B cells proliferate, producing two kinds of daughter cells. One is plasma cells, okay, which is responsible during the primary immune response. Another one is memory cells, which are responsible during the secondary immune response. Okay, as you all know, B lymphocytes responsible for the production of the antibodies, which is responsible for the primary immune response, which is very delayed period of immune response, whereas memory cells, which are very rapid type of response, which gives the secondary immune response. Okay, they'll have the memory. If the memory cells which encounter the same antigen for the second time, the immune response will be very rapid. Further, fifth question, inflammation. Inflammation is a signal mediated response to cellular insult by infectious antigens, toxins, and physical stresses. While acute inflammation is important to the immune response, chronic inappropriate inflammation can cause tissue destruction. That also brings autoimmunity, neurodegenerative, cardiovascular disease. Or this inflammation we can define as a localized physical condition in which part of the body becomes reddened, swollen, and often painful, especially as a reaction to injury or infection. Further, we can see in the inflammation acute phase, we can write about the agents, cytokines, then events, pro-inflammatory cytokines, then sequence, signs or symptoms also we can write it over here. Okay. Inflammation is variably accompanied by fever, we can say pyrogenesis, redness, rubor, swelling trigger, pain dolor, and tissue or organ dysfunction, that is functio lessa. So this is all about the 5 questions for 2 marks. Now let us go to the section B. Answer any 4 of the following. Each 4 questions carries 5 marks each. So sixth question, differentiate between humoral and cell-mediated immunity. Each differences carries 1 mark. So this question is for 5 marks. If you write 5 differences, you will get 5 marks. Now let us go to the humoral mediated immunity and cell mediated immunity. As you all know, humoral mediated immunity is nothing but the antibody mediated immunity, whereas cell mediated immunity is the immunity, a defense mechanism or protection against the infection caused by the cells. Now let us go through the differences. Humoral mediated immunity. Humoral immune response is also called B cell mediated immunity because B stands for bone marrow. Bone marrow lymphocytes are involved in this response. Okay. Then here you can go through cell mediated immunity. Cell mediated immune response is carried out by the T cells. That is nothing but the thymus cells or T lymphocyte. So it is also called T cell immunity. This type of immune response is to defend against pathogens that may invade host cells. Okay. The next difference here is, just a moment. Okay. Humoral immune response is to defend the body against pathogens that may invade body fluids or humor. Okay. Humor, that is nothing but the fluid. B cells are antigen specific. Okay. The B cells are very antigen specific, means whatever the antigen which triggered the B lymphocytes for the production of the antibody, the B cells will produce the specific which is going to react with that specific antigen by producing the specific antibodies. Whereas in cell-mediated immunity, the surface of the T cells has receptor molecule that can bind with antigens, and these receptor molecules are made of a variable unit similar to the variable portion of the humoral antibody. A single T cell has about 1 lakh receptor sites. Humoral immunity protects against extracellular pathogens. Cell-mediated immunity protects against intracellular pathogens. Okay. In humoral mediated immunity, during immune response, B cells specific for the antigen enlarge to become lymphoblasts that further differentiate to form plasma cells. The mature plasma cells produce gamma globulins or immunoglobulins called antibodies at a rapid rate of about 2000 molecules per second for each plasma cells. The antibody secreted into a lymph eventually enter the blood. Okay, when an antigen enters the body, the macrophages, which is this macrophages can also be known as APC. APC stands for antigen presenting cells, first attack the antigen and fragment it into pieces. It then presents a piece of antigen to the T helper cells. The T helper cells recognize the antigen and trigger off a series of cell mediated immune response. Okay, hope you can go understand here. You can keep in your mind this macrophages is one of the APC. This APC which is going to present a fragmented or digested pieces of antigen to the T helper cells to trigger further cell mediated immune response. Okay, the last difference is here. The antibody molecule is the basic functional unit of this type of immune response. So this immune response is also called antibody mediated immune response. Whereas in cell mediated immunity, a clone of T lymphocytes is first formed after being activated by the T helper cells. There are different kinds of T cells which are morphologically similar but differ functionally. Okay, for 5 marks this much information is more than enough. If you have understood from your teacher more information, okay, that you can also write it here, but 5 marks this much enough. If they ask this one for the 10 marks, that time humoral immunity for 5 marks and cell mediated immunity for 5 marks, that time you can write further more information about this B lymphocytes, and also here other different forms of T cells can also be written. Okay. The next question here, seventh question, electroporetic method of gene transfer. Okay. In this, when you write about electroporation, usually we'll differentiate the marks like this. Definition 1 mark, principle, uh, here 2 marks, then procedure and application 2 marks. This is how we are going to divide when you write the answer. If you write just a definition and the flow chart, you will get only the 3 marks. Applications also you must write. Okay, now let us go through what is this electroporation. Electroporation or electropermeabilization is the process of applying electric field to a living cell for a brief duration of time in order to create microscopic pores in the plasma membrane called electropores, and that will be done only for the transient period. It means only for temporarily, only to make those our foreign DNA to enter inside the cells. This technique is used for transferring the recombinant DNA molecules into animal cells or even to the plant cells. Okay, whereas this method is also one of the indirect method of gene transfer or vectorless method of gene transfer. Okay. Now let us go through the principle. The phospholipid molecules of the plasma membrane are not static. When we apply electric field to them, their kinetic energy increases, resulting in the increase in the membrane permeability at certain points. Okay, this is exactly where we see the formation of electropores. Very minute pores can be absorbed in the plasma membrane when we put the electric field. Okay, the recombinant DNA can pass through these transient pores before they close. Transient means that is temporary pores. As you could make out in this diagram of gene transfer by electroporation, this is the culture cells where you can take the cells over here, and this is the gene of interest we have taken with the selectable marker genes, or you can directly take the DNA molecules. Both will be mixed together in a specific cuvette, and we are going to apply the certain amount of the voltage. When you apply the voltage, what happens here? Okay, this is the chromosome, and this is the nucleus, and this is the host cell, and this is our DNA molecule. Okay, so this DNA molecule, when you put the voltage at this transient condition, temporary condition, you can see the micro pores can be seen. Okay, microplasma pores can be seen here. Once the pores formed, immediately these cells will be taken up by the DNA. The cells take up DNA through the micropores in membrane. Okay, only for that, when we apply the electric field, pores will form, okay, to that voltage, and later the pores will be closed, and that is taken up by the nucleus, where you could see the gene of interest within the nucleus of the DNA. DNA enters the nucleus, and there you can see the presence of selectable marker genes for further screening of the our desired DNA molecule. Then you can go for the apply selection. Some of the cells may uptake our desired DNA. Some of the cells they may not uptake the our DNA. Then that time no selection. Then transient expression cannot be possible. Whereas there, okay. Whereas in this case, when you apply the selection by using selectable marker gene, you can see the stable expression can be seen. Whereas when there is no selection, that time transient expression or temporary expression can be seen. That kind of temporary and non-stable expression that is not used in the rDNA technology. So this is about the gene transfer by electroporation in a very brief. Okay, being a science student, you must write this diagram by using the pencil and label. Okay, for 5 marks, that is more than enough. Now let us go through the procedure what has happened in the previous diagrammatic flow chart. In this process, cells are mixed with the recombinant DNA, and the mixture is placed in a small chamber with electrodes connected to a specialized power supply. Okay, then what happens? Brief electric impulse is discharged across the electrodes, which makes pores or holes in the plasma membrane. Okay, that's why it's mentioned electroporation. When you apply the electric field, you are going to create pores within the plasma membrane. These pores remain for some time and are again released themselves. Okay, sorry, resealed themselves. Recombinant DNA enters the cell during that pore, whatever transient pores which are formed that particular fraction of second. Okay, and later that enters, which are removed and plated in fresh selective medium for the further selection. Okay, the process of selection is then applied to isolate cells carrying recombinant DNA. So this is about the electroporation. Okay, okay, applications are missed here. You kindly make a note about the applications of the electroporation for 1 mark or 2 marks. Now let us go to the type 3 hypersensitivity. Definition 1 mark, mode of action 2 mark, and biological effect 2 marks. Okay, when an antigen enters within the body, then the body and the antibody reacts with antigen and generates immune complex, because this antigen which triggers the B lymphocytes or immune system for the production of the antibodies. That's why these antigen were known as antigen generator. Okay, anti stands for antibody, GN stands for generator. So these antigens are basically antibody generator, okay, responsible for the production of the specific antibody. This immune complex gradually facilitates removal of antigen by phagocytic activity of body. Large amount of immune complexes lead to tissue damaging, for example, that is type 3 hypersensitivity. For this reason, type 3 is called immune complex hypersensitivity. Now let us go through the mode of action. These reactions develop when immune complexes activate the complement system's array of immune effector molecules. Complement components like C3A, C4A, C5A split and produce anaphylaxis. which cause localized mast cells degranulation and increase local vascular permeability. Okay. Further, when formed bulky antigen antibody complexes aggregate and combine with the activated complement, they chemotactically attract the polymorphonuclear leukocytes. Okay, and these cells release lysosomal enzymes in large quantities to cause tissue damage. I request all the viewers, if I'm going very quick, kindly pause this video and make a note, and also you can go through the serial number of this particular slides. Further, let us go through the biological effect. The recipient of a foreign anti-serum develops antibodies specific for the foreign serum proteins from circulating immune complexes, and within days or weeks after exposure to foreign serum antigens, an individual starts to develop serum sickness, including fever, weakness, then vasculitis, that is rashes with edema, then erythema and lymphadenopathy, then arthritis and glomerulonephritis. Due to the deposition of IgG antigen complexes in the blood vessels cause local damage and deposit in blood vessels of kidney glomerule called arthros reaction. Inhalation of bacteria and fungal spores gives rise to a disease called formous lung-forming immune complexes in the epithelial layers of respiratory tract. Another type of hypersensitivity reaction is known as lupus, that is systemic lupus erythematosus. It is produced as a result of interaction of IgG and neuroproteins of the disintegrated leukocytes, that is autoantigens. Lupus is an autoimmune disease. Okay. Now the next question is passive and active immunization. Okay. Definition 2 marks, differences 2 marks, applications 2 marks here. Okay. Now let us go through the passive immunization. Passive immunization, in which antibodies against a particular infectious agent are given directly to the child or adult, is sometimes appropriate. These antibodies are taken from a donor and then processed, so the final preparation of contains high antibody concentration. At that point, they are given in the vein or by shot to the patient. Okay, passive immunization often used in children and adults who have weakened immune system or may not be good candidates for routine vaccination for other reasons. It can be used with people who have not been vaccinated against a disease to which they have been exposed. For example, the passive rabies immunization, that is rabies immunoglobulins, is commonly used after a certain type of wild animal bites a child. A passive immunization for hepatitis A gamma globulin may be helpful for people traveling to a part of the world where hepatitis A is common. They are typically given before children or adults leave on their trip. These are used less now that is there is a vaccine for hepatitis A. Okay, hope you understood here the passive immunization. Passive immunization here, antibodies which are produced in some other host system, and once the antibodies are produced, those antibodies will be given to the donor or needy where their immune system is very weak and are unable to produce the antibodies against the specific antigen. That time this passive immunization will be done like a instant immunization. Okay. Now let us go through the active immunization. Active immunization is the induction of immunity after exposure to an antigen. Okay, antibodies are created by the recipient and may be stored permanently. Whereas passive immunization is temporary, that is only given in the instant condition. Okay, for example, passive immunity, that is the breastfeeding Okay, only during that time, that particular baby will be nourished through the breast milk, where you could see the presence of secretory immunoglobulins. Okay. Okay, I have given here less information for active immunization. You write it over there. Okay, further more information about the active immunization. If I get time, I'll be doing separate video, particularly with this passive and active immunization. Okay, wait for me. The next question here, explain mechanical method of tissue disaggregation. Okay, definition 1 mark, methods 2 marks, and procedure 2 marks. Whereas, let us go through the mechanical disaggregation now, physical disaggregation. The tissues are carefully removed from a given spot, is aseptically removed when kept in a sieve of 100 sieve container. Okay, it is put in a sterile petri dish containing buffered medium with balanced salt solution. In short, you can say BSS. The cells are alternately passed through the sieves of decreasing pore size, 50 and 20 mesh. If desired, the process is repeated to get more disaggregation of cells. The debris remaining on sieves are discarded and the medium containing cells is collected. The cells are counted using a hemocytometer. If necessary, medium is diluted by serum to raise the level of cells to the 10 raised to 4 cells per ml. Okay. The other methods of mechanical disaggregation of cells are forcing the cells through syringe and needle are repeated by pipetting. Although the physical method is very quick and cheap, yet it damages many live cells. Here you could see the flow chart of the mechanical tissue disaggregation. First, we'll take the tissue sample, okay, then press through 100 mu m mesh, then petri dish containing BSS you need to use for the further extract, then cells plus here tissue debris, repeat above process, then pass through the 50 micrometer mesh. Once you get the more cells plus tissue debris, repeat above to process by passing through the 20 micrometer mesh, mu m mesh, then viable cells plus tissue debris, remove tissue debris here, count cells with hemocytometer. Okay, then viable cells were taken and damaged cells were removed, dilute medium up to 10 raised to 4 cells per ml, separate viable cells from the damaged cells and seed out only the 10 raised to 4 cells per ml, then incubate and you are going to get your primary adherent cell culture. Okay. Further, the viable dissociated cells are now termed as primary cells. When the primary cells are seeded on culture medium in high density, these grow well. Thus, the primary variable cells of primary culture are called adherent culture and the cells adherent cells. Moreover, at this stage, some of the non-viable cells, if growing, along with adherent cells, can be separated out by using the second medium. Similarly, primary culture can also be grown in suspension. Okay, in suspension, the non-viable cells are removed from primary disaggregates by centrifugation using phycol and sodium metrizote. Okay, in this way, viable cells are separated from non-viable cells. Okay, hope you understood here mechanical disaggregation of tissues. I request you to kindly pause this video or take screenshot and make a note in your notebook so that you'll get of actual information so that you can go through repeatedly to understand this particular answer. Okay, for 5 marks, if you write this much, answer more than enough. Now let us go through the 10 marks question, section C. The 11th question, that is A. Here I have given two parts, A and B. Calcium phosphate mediated gene transfer, co-precipitation. Okay, here definition 1 mark, principle 2 mark, procedure and application 2 marks. Okay, let us go through the answer. This technique is used for the transfection of plant and mostly also animal cells. The recombinant DNA is mixed with calcium chloride in a phosphate buffer as neutral pH. Okay, this results in the formation of recombinant DNA calcium phosphate complex, which appears as a thin precipitate. This precipitate is then added to the host cell. Okay, the precipitate is taken up by the cell by the process of phagocytosis. The recombinant DNA enters the nucleus and integrates into the host genome. The transfection efficiency can be increased by exposing the whole cell to 10 to 20% of glycerol or dimethyl sulfoxide, that is in short DMSO. Okay, keep this in your mind. Usually somewhere you get this particular question for 1 mark too. Okay, now let us go through the B. Here, see, I have missed only have given a definition, okay, and principle. Not explained about the precision applications. You must explain here the precision applications of this calcium phosphate method of gene transfer. The B next part in the same question, primary explant technique, definition 1 mark, principle 2 mark, explanation 2 mark. Here, the primary explant technique was in fact the original method developed by Harrison in the year 1907. This technique has undergone several modifications and is still in use. The simplified procedure adopted for primary explant culture is depicted in flowchart and briefly described in the next slide. Here you can make out, okay, the primary explant technique for the primary culture. First and foremost here, the tissue were taken in the basal salt solution, okay, that is the BSS in short, is finally chopped and washed by settlings. Okay, then the basal salt solution is then removed. Okay, the tissue pieces are spread evenly over the growth surface after the addition of appropriate medium. Okay, then incubation is carried out for 3 to 5 days. Then the medium is changed at weekly intervals, okay, until a substantial outgrowth of cells is observed. Now the explants are removed and transferred to your fresh culture vessel. Okay, you can make out this one, right? Tissue in basal salt solution finally chopped will be taken here, then wash by settling, okay, then remove basal salt solution, that is BSS, then tissue process pieces were kept in the incubation medium for the on the growth surface, then incubate and change medium at weekly intervals for the subculturing condition, then further explants are removed and transferred to a fresh culture vessel in the this is all about the primary explant technique for primary culture. The primary explant technique is particularly useful for disaggregation of small quantities of tissues, for example skin biopsies. The other two techniques, that is mechanical or enzymatic disaggregation, however, are not suitable for small amounts of tissues as there is a risk of losing the cells. The limitation of explant technique is the poor adhesiveness of certain tissues to the growth surface and the selection of the cells in the outgrowth. It is, however, observed that the primary explant technique can be used for a majority of embryonic cells, for example, fibroblast and myoblast, epithelial cells, glial cells. Okay, glial cells. Suppression of viable and non-viable cells. It is a common practice to remove the non-viable cells while the primary culture is prepared from the disaggregated cells. Okay, this is usually done when the fast change of medium is carried out. Okay, the very few left over non-viable cells get diluted and gradually disappear as the proliferation of viable cells comments. Sometimes the non-viable cells from the primary cultures may be removed by centrifugation. Okay, the cells are mixed with ficol and sodium metrizoate, and later that will be centrifuged. The dead cells from a pellet at the bottom of the tube can be observed. Okay. So next question is your complement system. Specifically here you can recall about the classical pathway. Okay. This question is for 10 marks, definition 2 marks, methods 2 marks, and explanation 6 marks. So total 10 marks. The complement system is a part of the immune system, consists of a series of proteins that interact with one another in a highly regulated manner in order to eliminate pathogens. Okay, it helps antibodies and phagocytic cells to clear pathogens and damaged cells, which promote inflammation and attack pathogens plasma membrane. Proteins that take part in the complement system are called complements that collectively work as biological cascade, the sequence of reactions, each being the catalyst for the next. Okay, further, Jules Bordet in the year 1895 identified complements as heat sensitive components in the blood bearing non specific antimicrobial activity. Okay. All right. Further, the classical pathway begins with the formation of antigen antibody complex, that is the immune complex. When an antigen enters the body, the antibody, that is IgM or IgG, binds to it initially. Okay. This induces conformational changes in the Fc portion of the antibody. Fc, that is stands for fragment of crystallization. Okay, portion of the, that is the bottom region of the antibody, which exposes a binding site for C1 protein. Hence, the antibody activates the complement system only when bind to an antigen. C1 is a large multimeric protein complex composed of one molecule of C1Q and two molecules of C1R and C1S subunits. C1Q binds to the antigen bound antibody to the Fc portion. Then C1R and C1S are proteases which help to cleave C4 and C2. The immune complex bound to C1 calls another protein, that is C4, which is cleaved into C4A and C4B. C4A goes away, whereas activated C4B attaches to the target surface near C1Q. Now, C4B attracts C2, which is also cleaved into C2A and C2B. C2A binds C4B, forming the C4B2A complex, whereas C2B goes away. The active C4B forming this C42A complex, whereas C2B goes away. Okay, the active C4B C2A activates C3. The C4B2A complex is also known as C3 convertase, as this converts C3 into an active form by separating C3A and C3B. One molecule of C4B2A can cleave a large number of C3 molecules. C3B binds to the microbial surface or to the convertase itself. So which is convertase here? C3 is the convertase. C3b, when binds to C3 convertase, forms C4b, C2a, C3b. Okay, in short, we can say C5 convertase, right? With which activates C5. C5 convertase cleaves C5 into C5a and C5b. C5a further diffuses away, but C5b is stabilized by binding C6. Then C5b, C6 binds to C7, then C5BC, 6C7 complex is then inserted into the phospholipid bilayer of the cell membrane, which further binds C8. Okay, these cells, that is C5B6, 7, 8, activate C9 to form a macromolecular structure called membrane attack complex, that is MAC. This makes hole in the bacterium. As a result, the intracellular contents leak out and unwanted substances get in. Thus, the cell cannot maintain its osmotic stability, and the lysis occurs by an influx of water and loss of electrolytes. This is more effective in gram-negative bacteria than in gram-positive bacteria because MAC, membrane attack complex formation, is easy in the outer membrane in gram-negatives, whereas it is very difficult in the rigid thick layer of peptidoglycan in gram-positive microorganisms. Okay, some of the C3b molecules do not associate with the C4b2A. Instead, these molecules coat immune complexes or microbial cell surfaces and work as opsonins. Okay, this process is called opsonization, in which opsonin molecule binds one side to the particulate matter. That is, in bacteria, tumor cells, RBC, and other side, they bind to the cell receptor of phagocytic cells like neutrophils and macrophages, which enhance the process of phagocytosis. Smaller complement subunit diffuse from the site and can initiate localized inflammatory responses by binding to specific receptors. Okay, that is about the complement system of the classical pathway. Now the next one is transgenic sheep. Definition 1 mark, principle 2 mark, production and applications 2 marks. Sorry, here production and applications 2 marks here. This is for 5 marks. It means in 1310 when question 10, 13 number question which carries 10 marks, where I have given the two question, that is A is transgenic shape and B is your barriers of innate immune system. Okay, so now let us go through the 13th question. A, answer transgenic sheep. Transgenesis experiments in sheep and goats mostly involve the development of memory glands, okay, as bioreactors for the production of proteins for pharmaceutical use. This is possible uh, de- despite that fact that quantity of milk produced by sheep and goats is less than that of dairy cattle, for example, cow and buffalo. Some proteins produced by sheep and goats have good pharmaceutical use. Okay, transgenic sheep with increased wool production. Let us go through that one. Whereas keratin is the wool protein with highly cross-linked disulfide bridges for good production of quality wool. The amino acid cysteine or its precursor methionine is required in large quantities. However, cysteine supply to sheep is always inadequate since the microbes harboring the rumen utilize it and release it in the form of sulfide. This problem can uh, overcome by producing transgenic sheep containing bacterial genes for the synthesis of cysteine. Okay, the two enzymes synthesized by the transgenes are capable of trapping the hydrogen sulfide liberated in the intestine to produce cysteine. Thus, good supply of cysteine to sheep improves the quality and quantity of wool. Now let us go through here barriers of innate immune system. Okay, you can also go through my previous video where I have explained in detail about innate immunity or non-specific immunity or native immunity. Okay. Okay, let us go through here. Barriers of innate immune system. Definition 1 mark, types 1 mark, explanation 3 marks. Okay, physical barriers. There are mechanical barriers to many microbial pathogens. There are two types of uh, immune system, that is one is innate immunity and the one is the acquired immunity. Okay, whereas in this physical barriers, you can recall about skin and mucous membrane type of physical barriers. The skin is physical barrier of body. It is its outer tough layer, the stratum Corneum prevents the entry of bacteria and viruses and other invading organisms. Okay. The second one, mucous membranes. Mucus secreted by mucous membrane traps the microorganisms and immobilizes them. Immobilizes means mobility, means their movement will be immobilized. They'll make that into static condition, or they will not allow. Microorganisms and dust particles can enter the respiratory tract with air during breathing, which are trapped in the mucus, right? The cilia sweep the mucus located with microorganisms and dust particles into the pharynx, that is throat, right? From the pharynx, it is thrown out or swallowed for the elimination with the feces. Further physiological barriers like the skin and mucous membranes secrete certain chemicals, okay, which they dispose of the pathogens from the body. Okay, for example, body temperature, then pH of the body fluids, and various body secretions which prevent growth of many disease causing microorganisms. Some of the important examples of physical barriers are as follows. Okay, acid of the stomach kills most ingested microorganisms. Bile does not allow growth of microorganisms. Okay, whereas cerumen, that is earwax, traps dust particles, kills bacteria and repels insects. Lysozyme is present in tissue fluids and in almost all secretions except in cerebrospinal fluid, sweat and urine. Then lysozyme is in good quantity in tears from eyes. Lysozyme attacks bacteria and dissolves their cell walls. Lysozyme is also found in saliva. Okay. Then further, nasal hair. They filter out microbes and dust in nose. Urine, it washes microbes from urethra. Vaginal secretions, it is slightly acidic, which discourages bacterial growth and flush microbes out of vagina. Sebum, which is the sweat, it forms a protective acid film over the skin surface that inhibits growth of many microbes. Okay. The next question is heart selection and screening of hybrid cells. Definition 2 mark, principal 2 mark, explanation 6 marks. Okay. Hybridoma, a selection after fusion of melanomas and spleen cells is critical step in monoclonal antibody, okay, production. Often scientists use in this case the hat, that is hypoxanthine aminopterin thymidine method to accomplish this task. During the fusion process, three types of cells are present: unfused myeloma cells that are deficient in an enzyme called HGPRT, unfused spleen cells, and fused hybridoma cells. Unfused spleen cells are easily selected against since they do not replicate in culture. Unfused myeloma cells can be selected against using media containing HAT. The aminopterin found in the medium blocks the de novo DNA nucleotide synthesis pathway. Typically, when the de novo pathway is blocked, cells will then utilize the salvage pathway as an alternative means to replicate only if hypoxanthine and thymidine are present. Okay. Further, however, these myelomas are unable to do so since they are deficient in an enzyme called HGPRT, which is required for the salvage pathway. Hence, myelomas are unable to replicate in culture. Only hybridomas survive. Okay, hybridomas inherit a functioning HGPRT enzyme from the spleen cell. So, even though the de novo pathway is blocked, they can still use the salvage pathway to replicate. Okay, hat medium selection. Hat selection depends on the fact that the mammalian cells can synthesize nucleotides by the two different pathways, that is the de novo and salvage pathways. The de novo pathway, in which a methyl formyl group is transferred from an activated form of tetrahydrofolate, is blocked by aminopterin, a folic acid analog. Okay. When the de novo pathway is blocked, cells utilize the salvage pathway, which bypasses the aminopterin block by converting purines and pyrimidines directly into DNA. The enzymes catalyzing the salvage pathway include hypoxanthine guanine, phosphoribosyl transferase, that is HGPRT, and thymidine kinase TK cells. Okay, a mutation in either of these two enzymes blocks the salvage pathway. Hat medium contains aminopterin to block the de novo pathway and hypoxanthine and thymidine to allow growth via the salvage pathway. When two types of cells, that is one with a mutation in TK and other with the mutation in HGPRT, are fused, only the hybrid cells will contain the full complement of necessary enzymes for the growth on hat medium via the salvage pathway. Thus, only hybrid cells will grow in hat medium. Okay, here you could see the normal T or B cells which dies after 7 to 10 days in culture. Okay, here the cancerous T or B cells grows continuously in culture. Okay, then poly polyethylene glycol will be used here. Then you can see the nucleus of normal lymphocyte and nucleus of cancer cells, that is in heterokaryon. Okay, random chromosomal loss can be seen over here. Then the hybridoma which are formed expresses some normal B cells or T cells genes but grows indefinitely like a cancer cells. Okay, here you could see the B cells hybridoma responsible for the monoclonal antibody and T cell hybridoma responsible for the interleukin 2 specifically. Okay. The next question and the last question in the 10 marks is antibodies, that is IgM and IgG. Okay, now let us go through the antibodies IgM and IgG. Definition 1 mark, labeled diagrams, explanation 6 mark. Okay, okay. Definition 1 mark, labeled diagram. How much marks here? 3 marks. Then explanation 6 mark. Okay, now let us go through very quickly. Antibodies and structure of IgM and IgG. Explanation: antibodies are immunoglobulins. In short, we can write Igs. When you write this Ig, when you represent the structural condition, then you are going to write this as Ig immunoglobulin. When they comes to the functional aspects, you'll use the terminology antibody, which are produced in the body in response to the antigen or foreign bodies. Thus, all antibodies are immunoglobulins, but all the immunoglobulins are not antibodies. The antibodies are found in the serum fraction of the blood and are also known as immunoglobulins, that is Ig. The chemical composition and structure of antibodies were revealed by G.M. Edelman and R.M. Porter, who received Nobel Prize in the year 1972 in physiology and medicine for this particular contribution. Okay, if you want to go through separately about antibodies, I have given the videos specifically for the structure of antibodies and specifically all different types of immunoglobulins have explained in detail. Kindly have a look. Now let us go to the 5 different types of immunoglobulins, okay, where you can keep this in your mind as game D, GAMED, game D, okay, or GAM D. Name of the immunoglobulins IgG, A, M, D, E. Nature, based on the nature of the heavy chain, we have classified all these immunoglobulins. Okay, gamma, alpha, mu, delta, epsilon. Subclasses or subtypes in IgG, we have IgG1, IgG2, IgG3, IgG4. Whereas in IgA, we have only IgA1 and IgA2. We don't have further subclasses in all these immunoglobulins types. IgM, this is pentameric, but that is not a subclass. Okay, that is a single entity, that is IgM. Okay, this is the general structure of the immunoglobulin. Okay, where you could see the presence of the light chain. Okay, this is the antigen binding region. Okay, and here you can see the variable light chain. This is the disulfide bond, and this will be known as the one domain. Okay, this is the constant light chain. Okay, and this is the variable heavy chain. Okay, this is the variable heavy chain domain, disulfide bond. What kind of disulfide bond? Intra disulfide bond within. whereas this is the interdisulfide bond. Okay, this is the constant heavy chain first domain, interdisulfide bond, whereas these two are the inter between the two polypeptide chain disulfide bond, interdisulfide bond, and this region is known as the hinge region, okay, which is holding the two heavy polypeptide chains. Okay, here you can see the presence of carbohydrate fraction is very less amount can be seen. This is constant heavy chain first domain, constant heavy chain second domain. This is constant heavy chain third domain. Okay, and here you can see, you can call this as the FC region, and this will be FAB region, that is fragment of antigen binding region, and this is the FC region of the immunoglobulin. Whereas here you can see this is the N terminal region or amino region, and this is the carboxyl region. Please go through my previous video. I have explained in detail about the structure of the immunoglobulin. Now let us go through the all the different five immunoglobulins and their functional concept. IgA, where you could see the presence of two subclasses, IgA, right, A1 and A2, found in mucous areas such as the gut, respiratory tract, and urinogenital tract, and prevents colonization by pathogens, also found in saliva, tears, and breast milk. IgD functions mainly as an antigen receptor on B cells that have not been exposed to antigens. It has been shown to activate basophils and mast cells to produce antimicrobial factors. IgE binds to only one immunoglobulin, binds to allergens, one class, okay? Um, Binds to allergens and triggers histamine release from mast cells and basophils and is involved in allergy. Also prevents against parasitic worms. IgG, we have a four class, that is IG G1, 2, 3, and then 4. In its four forms, provides the majority of antibody-based immunity against invading pathogens. The only antibody capable of crossing the placenta to give passive immunity to the fetus is the IgG immunoglobulin molecule. IgM, only one class, which are expressed on the surface of B cells, okay, which is in a monomer and is and in a secreted form that is pentamer with very high avidity. Eliminates pathogens in the early stages of B cell mediated immunity, that is humoral mediated immunity, before there is a sufficient IgG immunoglobulin molecule. Okay, one more you can keep in your mind. IgD is performing like a membrane bound immunoglobulin. Okay, along with that, even IgM is also one of the membrane bound immunoglobulin. Now let us go through about IgM. IgM is about 5 to 10% of all antibodies in the blood. It is also found in lymph. It is the largest immunoglobulin which secretes first by the plasma cells. Okay, this is responsible for the primary immune response. It is so named because it is a macroglobulin at least 5 times larger than the IgG, and M stands for mu. IgM is the oldest immunoglobulin class, our earliest immunoglobulin class. It activates the B cells. It is also the earliest immunoglobulin to be synthesized by the fetus. IgM has a J chain, and its each dimer contains polypeptide called a secretory component. Okay, that like a S shape. It cannot cross the placenta barrier. Okay, IgM is 500 to 1000 times more effective than IgG in opsonization, that is described to be height, or I explained in the previous video also, you can go through in bacterial action and in bacterial agglutination. But in neutralization of toxins and viruses, it is less active than IgG, but it helps in the complement activation. Okay, if I'm going very quick, kindly pause this video and make a note. This is the structure of the IgM immunoglobulin molecule, which is a pentameric form of IgM, where you could see here the J chain. Okay, that is the J chain can be seen here. Each immunoglobulins which are connected by the disulfide bonds. What is the speciality here? Instead of presence of only CH3 in IgM, you can see additional domain. Constant heavy chain first domain, constant heavy chain third domain, constant heavy chain third domain, and is a constant heavy chain fourth domain. Okay, this is the overall view of the IgM structure, that is pentamer. A percentage serum antibodies 5 to 10%, location blood, lymph, B cell surface, and monomer. Half-life in serum 5 days, complement fixation yes, Placental transfer, no. Known functions, first antibodies produced during an infection or earliest immunoglobulin which is produced against the infection antigens. Okay, effective against microbes and agglutinating antigens. Okay, you can also brief here IgM, which is the third most immunoglobulin, which account for 10% of the total immunoglobulin pool, largest immunoglobulin here. Okay, often referred to as the macroglobulin because of the molecular weight 950 kilodaltons and monomer with molecular weight of 180 kilodaltons. Each immunoglobulin with the two light chains that is known as kappa or lambda and two heavy chains which are of mu type. IgM accounts for 5 to 10% of the total immunoglobulin. In IgM, you can see the polymers of 5 molecules that is pentameric which are composed of 5 subunits held together by disulfide bridges at carboxy terminals ends of the mu heavy chain. Half-life is 5 days. They have 10 combining sites per molecule which can act or interact with each other on the same molecule when it binds to microbes. Because we have 5 immunoglobulins, each immunoglobulin is 2 fragment of antigen binding sites. So you'll be having 10 combining sites. Thus, overall tightness of binding of an IgM molecule to microbe avidity is quite high, making antibodies very effectively to remove microbes. Okay, this pentameric IgM contains an additional polypeptide chain and is necessary for the polymerization of the monomeric IgM to form pentameric IgM. IgM is the first antibody produced by the lymphocytes to appear in the primary immune response to an antigen and expressed on the surface of B cells, which acts as an antigen receptor of these cells acting like a membrane bond. IgM is found primarily in the intravascular space and is first antibody produced in an immune response. Okay, IgA is the major antibody found in secretory fluids, but IgM plays an important accessory role as secretory immunoglobulin. It is the earliest immunoglobulin synthesized by fetus, okay, synthesized during the third month of gestation, which are effective against toxins like diphtheria, tetanus, anthrax, and many more. Here you could see the production of the antibody. During the production of the antibodies, we are going to introduce the different doses of antigen to the laboratory animals. For example, sometimes the low molecular weight antigens will be known as the haptons. Haptons in Greek word that is the hapten that is to fasten. Okay, whereas for to the non antigens are not basically a true antigens. To that we are going to use the carrier molecule like haptons, which is going to enhance the molecular weight of that particular antigens to trigger the immune system for the production of the antibodies. Here you could see this is the carrier protein with the haptons. Haptons are used as epitope. Okay, here when alone and haptons are used, there is no antibody produced. When these haptons, which are of low molecular weight, when they are tagged with the carrier protein with the hapton, that time there you could see the production of the antibody, that is anti-hapton antibodies can be seen. So now let us go through what are haptons. Haptons are the small molecule that elicit an immune response only when attached to a large carrier such as protein. The hapton carrier complex in turn stimulates the production of antibodies which the unbound hapton cannot do and becomes immunogenic, that is capable of eliciting the immune response. For example, you can use hapton as aniline, para-amino benzoic acid, any other carbohydrates, proteins can be used. Now let us go through the biological functions of IgM. IgM is the natural antibodies which are residing in the A blood group, B blood group, O blood groups and are of IgM class. It does not cross placenta, but human fetus can synthesize IgM if its B cells are antigenically stimulated as in congenital infection. Congenital, that is by birth. It is the first antibody to appear in the primary immune response by it does not persist for long because if it's short of life, therefore it is an useful indicator of recent infections. It means if there is a more amount of the IgM for longer period, that you can see the body got infected for the that particular situation. IgM having multiple antigen binding sites, it inhibits the mobility of pathogens and enhance their phagocytosis. It also shows the properties such as opsonization and complement fixation. Because of macromolecular size, it is primarily localized in blood and thus protects against the blood serum infections. Now let us go through the IgA. IgA, it is the second most abundant class, constitute around 10 to 15% of antibodies of serum. Okay, it is mainly found in sweat, tears, saliva, mucus, colostrum. Colostrum is the first milk secreted by a mother, and okay, gastrointestinal secretions, you'll be able to see the presence of IgA. IgA in smaller quantities are present in blood and lymph. IgA has an extra polypeptide chain called J chain or joining chain, and extra protein chain known as secretory component. Levels decrease during the stress, lowering resistance to infection, provides localized protection in external secretions. For instance, tears, intestinal secretions, etc. Against bacteria and viruses. Okay, IgA is excreted through feces. It is also called capro antibody. This is the structure of the immunoglobulin IgA, dimeric form of a secretory immunoglobulin A, where you could see the presence of IgA monomer, here another IgA monomer, which are held together by the secretory component, S-shaped secretory component, basically polypeptide chain, and also stabilized by the joining chain, that is the J chain. To summarize about IgA structure, which is a dimer, percentage serum antibodies 10 to 15%, location that is in the secretions like tears, saliva, intestine, milk, and also they are found in the blood and lymph. Half-life in serum is 6 days. Complements fixation cannot be seen. Placental transfer cannot be seen. Known functions, localized protection of mucosal surfaces and provides immunity to infant digestive tract. This IgA which can also recall in the passive immunity, okay, because newborn babies will be protected by the breast milk, that is the passive immunity to the newborn baby. Okay, IgA, which is, to again summarize, IgA is the second most abundant class, which accounts for 10 to 15% of antibodies or immunoglobulin pool. Okay, this is the second most immunoglobulin class, which are found in sweat, tears, saliva, mucus, colostrum, and first milk secreted by the mother can also be seen, and gastrointestinal secretions also can be seen. Saliva, that is IgA concentration is 70 to 80%. Smaller quantities are present in blood and lymph. IgA, an extra polypeptide chain called J chain can be seen, and extra protein known as secretory components can be seen. See, I've given more information here, have some of the information repeated, but you can use any certain slides for your examination purpose. This IgA, which provides localized protection in external secretions like tears, intestinal secretions, etc., against bacteria and viruses. When IgA is excreted through feces, it is called capro antibody. In serum, IgA present in the form of monomer of two heavy chains and two light chains. Okay. Most of IgA occurs in the serum as a polymerized, and later that will be a dimerized, okay, joined together by a polypeptide J chain, and later it has a dimeric condition. It has four polypeptide chains, two light chains, that is of kappa or lambda, two heavy chains are of alpha type, which molecular weight is on 1,60,000 Daltons, half-life 6 to 8 days. J-chain component facilitates polymerization of both serum IgA and secretory immunoglobulin A. IgA special features, that is associated with secretory mucosal surfaces. Okay, IgA when transported from mucosal associations, lymphoid tissues to mucosal surfaces, it acquires the polypeptide termed as secretory protein called a secretory antibody. The secretory piece stabilizes IgA, when transported from mucosal associated lymphoid tissues to mucous surfaces, which acquires polypeptide termed as secretory protein called secretory antibody. Okay, and the secretory piece stabilizes the IgA dimer against proteolysis. Okay, that is from protease enzyme and is single polypeptide chains of molecular weight 70,000 Dalton, that is secretory component chain. IgA synthesized by plasma cells and lamina propria, that is the thin layer connective tissue of mucosa, of mucous membrane, and dimerized intracellular by secretory chain and J chain to form dimers. Now let us go through the biological functions of IgA, immunoglobulin. Okay, this acts as an anti-septic mucous membrane. Okay, it binds to the pathogenic microorganisms and it prevents the attachment of pathogens to the mucus of respiratory, gastrointestinal and urinogenital tract. This provide immunity against tapeworms, okay, which are found in colostrum, breast milk, which protects the baby from intestinal pathogens and also helps to protect and nourishing newborn. In the intestine, secretory immunoglobulin attaches to viruses, bacteria, and protozoans, parasites such as Entamoeba histolytica, which prevents the adherence to mucosal surfaces and invasions of host tissues. Okay, whereas you can also keep in your mind about the colostrum. What is colostrum? Colostrum, which is a thick yellowish fluid produced by breast in first few days after the birth of the baby and later replaced by mature milk. It suits the child because less fat and sugar and rich with vitamin A, okay, and immunoglobulins which help the child against certain infections, okay, that is known as the colostrum, okay. This colostrum also protects poliovirus, streptococci, diphtheria, tetanus, mumps, and rubella, okay. That is about the colostrum. I have not written here colostrum. Please keep that in your mind. In colostrum, you'll be able to see the presence of IgA secretory immunoglobulin, which is going to give the passive immunity to the newborn baby. Okay, secretory immunoglobulin A, which is going to provide the passive immunity, and which is can also be known as naturally acquired passive immunity or maternal immunity, or the mother provides resistance to a child by direct transformation of antibodies, that is to the colostrum, that is the milk, okay, that is like an instant immunity. They neutralizes local toxins by promoting phagocytosis process and various other intracellular pathogens that reside within the epithelial cells. Secretory immunoglobulins found in tears, saliva, and breast milk and forms an immune barrier and plays a major role in protecting surface tensions and against infection. Hope this is about the IgA immunoglobulin. Hope you have understood. This is all about the secretory immunoglobulin IgA. Okay, I have given much information here. You can cut short and you can write about the both immunoglobulins IgM and IgA. Okay, now let us go through here about section D. Okay, here section D is you are going to write for 1 mark answer. Okay, section D, the last slide you'll be going through with the 1 mark question answers will be going through here. In section D, the questions asked were uh, one marks each. You can write in one sentence. Okay, for example, section D, they've asked your cell strain. A cell strain which is derived either from primary culture or a cell line by the selection or cloning of cells having specific properties or characteristics. Okay, 17 question, transgenic sheep. Okay, whereas in this case, transgenic sheep, the trans genes introduced into sheep inserted randomly in the genome. Adjuvants, adjuvants were originally described by Raman as substances used in combination with a specific antigen that produced a more robust immune response than the antigen alone, thus encompassing a wide range of materials. Okay, this adjuvant which enhances the immune response along with the antigens. Plasma, that is blood plasma, is a yellowish colored liquid component of blood that normally holds the blood cells in the whole blood in suspension. Okay, blood plasma, which is also you can say liquid part of the blood. Who discovered blood grouping? Blood grouping was discovered by Carl Landsteiner. Okay, so this is about the 1 mark questions. Okay, next one is fibronectin. Fibronectin is a high molecular weight, which is a 440 kilodaltons, which are a glycoprotein of extracellular matrix, which provides a cell adhesion molecule by anchoring cells to collagen or proteoglycan substrates. Okay, the next question is 22nd one. Opsonization is a term that refers to an immune process where particles such as bacteria are targeted for destruction by immune cells known as phagocyte. Okay, the process of opsonization is a means of identifying the invading particle to phagocyte. Okay, 23rd question here, that is HGPRT. The full form is hypoxanthine guanine phosphoribosyl transferase. Hope you'll be able to keep this particular answer in your mind about the HGPRT. Okay, the next one is 24th question, that is attenuation. Attenuation takes an infectious agent and alters it so that it becomes harmless or less virulent. Okay, 25th question, raft method. Raft method is that in this method, explant are placed on raft of glass or lens or paper or ran acetate, which is then allowed floated serum in a watch glass. Okay, hope you understood all these question and answer, right? That's it about the this particular question paper for 70 marks. Okay, further I have given here additional information like epitopes. Epitopes are the sites in the antigen molecule that bind to corresponding antigen antibody antigen binding sites of the antibody. Okay, T cell receptors are the convex regions in the antigenic molecule which bind to complementary concave regions in the antibody molecules called paratopes. Okay, valence, that is the number of antigenic determinant sites present in the antigen molecules, is called its valence. So this is about the epitope, paratopes, and valence. Okay, whereas in this particular case, I have mentioned here the flow chart of the cell systems, okay, of WBCs and RBCs and other origin. Okay, for example, you can make out here, this is the hemopoietic stem cells in the bone marrow, which is from this hemopoietic stem cells, you can see the stem cells for all except the lymphocytes in the bone marrow can be erythroblast, megakaryoblast, then myeloblast, then monoblast can be seen here. And further, you can see here the formation of red blood cells, erythrocytes, then platelets, then eosinophil, basophil, neutrophils, then monocyte later that will be differentiated into macrophages, and further dendritic cells, which are mononuclear phagocytes, right? Then further here, from this hematopoietic stem cells, you can see the lymphoid stem cell, natural killer cells, lymphoblasts, then further from the lymphoblasts, T lymphocytes and B lymphocytes, and these B lymphocytes responsible for the plasma cells. Okay, you can take screenshot or you can pause this video and make a note. Further, I have given here the different biological effects of antibodies. I have given here, you can go through here, generation of oxidants, then immunomodulation, opsonization, it brings, then activation of complement, then virus and toxin neutralization will be carried out by the immunoglobulins, antibody dependent cell cytotoxicity, direct antimicrobial activity, then reduced damage to host from inflammatory response. This is all about the different biological effects of antibodies or immunoglobulins. Okay, then inflammatory response, I've explained this in even in the immunology video to brief this inflammatory response, which is a localized physical condition in which part of the body becomes red and swollen or hot often painful, especially as a reaction to injury or infection. Okay, here you can see the stepwise inflammatory responses can be seen over here. Okay, I explained this concept in the history of immunology. Kindly go through that one. Okay, further, this is about the mechanism of phagocytosis. If you take a screenshot and if you go through all these steps, stages in phagocytosis process, that is more than enough for your examination point of view. Okay, hope you understood today's concept about immunology and animal biotechnology paper. Okay, so here I've explained today in this video the paper of 2018, that is the fifth semester examination, November December 2018. Have given the entire question answer here. Kindly go through and make a note of all the answers so that you can finish with one particular paper for 70 marks. Okay, I hope you understood today's class. I, Dr. Pratibha K, signing off for today. I'll be back with some more videos. Kindly stay tuned with me. And if you have not yet subscribed this channel, kindly subscribe, like, share, and do comment, okay, if possible. And send this video to the needy student, and that will be useful for them during this end COVID-19 pandemic situation. Thank you everyone for watching this video.